it's a real pleasure. And we have heard quite a bit about how data can be generated and what we can do with hardware and technology systems. But now we want to change gear a bit and take a different perspective. And in the next 15 to 20 minutes, I want to show you what can be done with the data, because we often say that the systems have potential, that it can revolutionize things. But this is a train that hasn't started rolling, and we try to give an answer to the question why. So, who are we? Company mentioned at the beginning. I chair the business unit of MPSlabs. They deal with the digital twins. This is embedded in MPSlabs. This is a bigger company. They do biosimulation in general, simulating pharmaceutical or pesticide compounds in animals or human beings. We are 35 plus employees, finance, business, physicists, chemical biology. So we have a broad representation of people in our company. And I see it as very positive because I have a family that we work from home. We are digital nomads, and depending on whether or not you like it, it's good if you don't like it. But MPSlabs and what we do with MPSlabs is that we help our customers, crop science, agriculture, cosmetics companies, in their decision-making processes to make the right decision at an earlier point in time than they've done so far. And we have different projects that we carry out at national, international level. And as we are a digital company, we focus on strategic partnerships with companies, and we depend on them as we don't generate data ourselves. So what do? We are active in 3 different areas: life science, R&D, hands-on research, like at university. We develop computer models for new diseases that are unprecedented, and we try to make a decision with as little data as possible. And on the other hand, we can personalize things and help pharmaceutical companies in their drug development process, and they can then use computer models and make decisions regarding safety or use of drugs in a population or subpopulation, like children, for instance. Then there is a third pillar, and that is that the software we use is open source and open access. There are no license fees, no royalties we pay, and we support in the further development of the software so that we can have a better offer for our customers. We as MPSlabs focus on 3 pillars. So 3 approaches, basically, where we see that with our computer models, people can get more out of them. First of all is architecture and design of new chips. If you want to design a chip from scratch, we, with our models, could help to answer the questions about the size that the chip needs to have, the cell culture mediums, the numbers. If you need a liver and an intestine and a brain chip, if you want to combine them, how do they need to be connected so that it works from a human physiological point of view, and so that it's relevant? And we mentioned AI, and I would like to distance myself from that. We don't work in a black box manner. Everything we do can be reproduced, is interpreted, an interpretation is possible. In the second pillar, we focus on how the data that's generated in the lab, how can it be assessed? and implement and interpret. And this is the part Ärzte is specializing in. That is the output of the second pillar. Data is generated as an output, which can be used in a computer to simulate how a drug will behave, whether there will be toxic side effects, and before you test it in human beings. In the next couple of minutes, I'd like to present two case studies. First of all, my point of view, and I mentioned it already, it's a non-animal drug development. Is it possible? Here you see a simplification of it. We saw that before. Drugs are researched, are then tested in animals, and then applied on people. The problem is that it's not functional. Several millions of animals are killed every year in the EU, and the success rate is roughly 10%. And and 2 billion euros are wasted, or 2 trillion rather. And if you showed this to an investor, they still wouldn't invest, because this is the state of the art that has happened for over 60 years. But now we have the microphysiological systems, spheroids, it's all pointing to the same direction, mini organs. And the mini organs have the potential to put drug development upside down. But the question that's still not answered, and that we want to answer, is how these results can be translated from the lab into human beings, so that we, at the end of the day, we have a solution that is more effective. And in order to do that, we have two solutions that we can offer. A kinetics part and a toxicity part. The kinetics part, here we take data from the MPS trial, and the data is entered into the mechanistic digital twin. Sometimes we also add some machine learning on top of it. But then with that, you can predict the pharmacokinetics, and in the other case, we analyze the toxicity, and we can predict the toxicity in drugs. What does it look like? The first part is our service, our DigiLox. It's a digital liver-on-chip simulator. Just to give you some background, that's nothing new. Drugs have been tested using liver cells for decades, but what we see across the board, and the new systems cannot change this against the promise that was made. You see a systematic underprediction of the clinical metabolization rate. That is the first parameter you apply to see how fast the substance is cleared in the human being, and this is very decisive for toxic side effects as well. And the question is if this systematic underprediction can be improved. And we chose an approach that we call the digital twin approach. We look at the biological processes that are there, which one we want to depict, what kind of information of the hardware, of the chip itself, but also the physical, chemical properties of the substances that we need to analyze. We put it all in a computer model, and we have this box representation of the physiology and the biology on the chip. The big advantage is that we have the maximal information that we have that we can use, and we can separate the processes which in the analysis so far had been put together. In order to give you an illustration, I'm going to show you some results. I would like to lead you through this slide, although there's a lot of information on it. So we looked at more than 70 drugs with our approach. In blue, you see the ratio between the clinical metabolization rate, the actual one, as compared to the prediction if you use the traditional approach. Here you see the underprediction. and you have a broad spread. For a new medication, new drug, that means that it's not clear what the clearance rate is. Do you overestimate it or underestimate it? With our approach, two things happen. A, we can shift the curve to 1, the mean. So our prediction is in line with the clinical results, largely, and we can also reduce the spread. This means that what we predict, we're quite certain that it's accurate. You see a breakdown of the same data with a box plot. CN Bio, Javelin, and spheroids. These are different chip manufacturers and liver cell culture systems. And independently, we see that we can improve the prediction, but we can also make statements about the performance or the power about the accuracy. So this allows us to benchmark the accuracy of our system against the others. That's not always nice for the chip manufacturers if they are not at the top of the list, but for the other companies, it's very important to have this information because they have to decide about the chip they want to buy, actually. I hope that this video plays. Probably not.
Dann nur so viel dazu. Wir haben eine App, die ist natürlich nicht so toll wie das, was man in dem App Store irgendwie runterladen
which is not as great as what you can download in the App Store, but it's a graphical surface that shows you quite easily the behavior and the breakdown. I can explain this also in the breakout room. We'll see if the video plays there.
Wenn man jetzt aber sozusagen die Vorhersage aus der Klinik in die Klinik besser gemacht hat, dann ist es am Ende des Tages.
After improving the prediction, it's still just a number. Still, we don't know what that means for the patient. This is something we try to present here on a different slide. What you see, the key messages are the pink dots. This is clinical data. If you administer a drug and then you test the blood over time, this is the curve you get. In blue, you see a computer model that we use to show that we have the data. But if you take the same data from a liver chip with a normal mathematical approach, you see that this is the red curve with the corresponding statements and explanations. If we use the same data set with the digital twin, this is the black curve. As a result, we can describe the data in a much better way. That's one result. And second, we could also forecast that the drug will be cleared from the human body in 24 hours. That's important because if you give the drug again after 24 hours, oftentimes you take the drug once per day over several days. Here we would simulate an accumulation of the drug, and that could have consequences for the evaluation of toxicity of a drug. One thing we also managed, as part of our company history, we were nominated for the Innovation Prize of Lower Saxony. We were nominated at the shortlist out of the first 3. We didn't win the prize, but this shows that ideas that don't make it to the top of the list can still find recognition and praise. And if you have an idea, it's also, even if it's not mature yet, fully mature, it's worthwhile spreading the news about it. And it's also been a great event to be part of. I hope I have 2.5 more minutes left, and then I will be done. I'd like to focus on toxicity again. We looked at kinetics, and now we want to look at toxicology. We heard about cardiotoxicity too. Here it's a typical curve, concentrations, and then you look at IC50. This is the situation when 50% of the biomarkers is still relevant. This is usually a curve like that. And then you compare it to clinical data. Here you did it with an emulates chip to predict liver toxicity. And this is an example for false negative.
Wir haben uns jetzt aber dieselben Daten
And
angesehen,
we looked at the same
haben das mit
data
unserem digitalen Zwilling
with our
simuliert,
digital twin.
haben
We
gesagt, hey, okay,
simulated it and we said, okay,
das Medikament kann
the
sozusagen an Zellen
drug
binden, aber eben auch
can
an das
bind to
Plastik
cells, but
von
also
der Chips. Es gibt einen
to the
Transport
plastic of the
des Medikaments
chips.
aus dem Medium in
The
die Zellen rein und wieder
drug is
zurück.
transported into the
Das muss alles
cells and
berücksichtigt
back.
werden.
And
Wenn
this
man
needs
das
to be
tut,
taken into
dann
account.
gibt
And if you
man
do this,
Korrekturfaktoren,
you get
die dafür
correction
sorgen, dass man diese
factors
rote
that
Kurve
make
nach
sure
links
that the
schiebt
red curves are shifted
zu
to the left
kleineren Werten, was
to
das Medikament
smaller
toxischer macht,
values, which
als es
makes the drug
bisher
be more
klassifiziert
toxic
wurde.
than
Und
it
darum
has
konnten wir
been
sozusagen das als True
classified so
Positive
far. This is why we could
nachklassifizieren.
determine
Das
it
Ganze
and
ohne
reclassify it as true positive.
neue Medikamente, neue
And
biologische
that
Experimente.
without new biological
Und jetzt
experiments.
noch eine letzte Slide. Ich habe
So
einiges
one
erwähnt.
last slide, I mentioned it at
Um
the
die wirkliche
outset.
prädiktive Power zu
In
zeigen,
order to
fehlt
really
es ja
show the
immer so ein
predictive
bisschen,
power,
ja, was bedeutet das denn wirklich? Also wie kann man diese
what we
Daten
need
in
to
den
prove is
klinischen
what
Effekt
does that
wirklich
actually mean?
übersetzen?
What is the
Und wir
clinical
haben das hier an
impact
zwei
of
Sachen
this
versucht,
data?
sozusagen einmal wirklich
And we
einfach zu
try to
berechnen.
do it with
Also
two
gehen wir davon
things
aus,
to
120
calculate it.
Firmen etwa 100
Let's assume
Medikamente
that
testen
20
wollen, um die
companies each want to
klinische
test 100
Kinetik
compounds
vorherzusagen.
to predict the
Das wurde mal an
clinical
Tieren gemacht, was
kinetics.
aber eine
It's
Unterprädiktion
done in animals,
zur Folge
which
hat.
leads to an
Da gehen etwa
underprediction of
80.000 Menschen
clearance,
drauf. Man
and
weiß also aus
80,000
biologischer
animals
Sicht,
die.
dass die Vorhersage
This,
schlecht
you
ist
know that
und
the
trotzdem
prediction will be
tötet man
poor,
80.000
but
Tiere.
still you
Können
kill
sozusagen,
80,000
das ist
animals
relativ,
for
das
basically
kann man
nothing.
nirgendwo verkaufen.
You can
Mit unserem
sell that
Ansatz
nowhere. But
und
with
mit
our
den
approach,
Chip-Daten sozusagen
with
und
the chip
einem
data, so to
Computermodell
speak,
des Menschen können wir
and the
sozusagen
computer model of
eine
a
almost spot-on
human being,
Prediction machen,
we
was
can
jetzt übertrieben
achieve an
Zahnarzt
almost
null
spot-on
reduziert. Das ist
prediction,
natürlich die
which
Realität leider
almost reduces
nicht
the number
ganz
of
null.
animals to
Jedenfalls
zero.
können wir
It's
die
not
Zahl deutlich
zero in
reduzieren
reality,
mit
but we
diesem
can
Ansatz, der
clearly
jetzt
reduce the
heute
number with
sozusagen
our
verfügbar
approach,
ist.
which is available already
In der
today.
Toxikologie sieht das Ganze noch ein bisschen anders
In
aus. Hier habe ich
toxicology, it
jetzt
looks slightly different.
Einfachheit halber
For the
nur
sake
eine
of
Company
simplicity, I
genommen, die 100
only
Compounds
selected
testen
one
will.
company testing
Normalerweise
100
haben
compounds.
Tiere ungefähr
Usually
50%
tests have
Sensitivität. Das heißt,
50%
gehen wir
sensitivity.
5000
This
Tiere drauf,
means
dafür,
that
dass man eigentlich eine Münze
5,000
werfen
animals
kann,
will
um
die
rauszufinden,
for a 50-50
wie
likelihood.
gut
You
das
can
Medikament
actually
ist,
use a
toxisch oder
coin to
nicht.
see if the
Mit
drug
Chips
is
kriegt man das auf
uh,
77%.
toxic or not.
Da sterben
With
aber immer
chips,
noch
you can
5000,
achieve
weil das
77%,
Ganze
but
nur
still
retrospektiv
5,000
ist.
needs to
Für
die,
eine
need to
richtige
die because it's retrospective.
prospektive Studie, wo man
If you want
keine
to have
klinischen
a
Daten
truly
hat,
prospective
könnte man
study
das
with
auf
no
irgendeiner Tube
clinical
sozusagen
data, you
reduzieren.
could
Das ist
reduce
sozusagen
it to
dann die
500
Phase 0,
animals.
wahrscheinlich das Muster, also diese 23%. Dann
These are
bräuchte man aber immer
those,
noch ein Computermodell
that's
des
the remaining
Menschen, um das
23%.
Ganze
You would
zu
still need a
simulieren.
computer model of the
Wenn
human
wir
being
das
to
auch noch mit
simulate it.
dazu nehmen und unseren
And if we
digitalen
add
Zwilling, dann können
that
wir das auf
and our digital
95%
twin, we
hochbrauchen,
can
was am
raise it
Ende
to
des Tages nur
95%
50 Tiere
sensitivity,
reduziert.
which means that at the end only 50 animals need to die.
Genau. Damit, um das Ganze sozusagen abzuschließen, das
And to
ist
round this off,
ein erster
to
Ansatz, um
conclude,
wirklich
this
nachzurechnen und
is the
wirklich
first
sich
approach
also mit der Mathematik hart hinzusetzen
to
und zu sagen,
really
das
do
sind die
your maths,
Einsparmöglichkeiten,
sit
die
down
es
and say,
gibt.
this is the
Ähm,
savings potential that there
und das
is.
ist das, was das Feld aus unserer Sicht sozusagen natürlich braucht,
And this
um
is what is
wirklich
needed in the
auch die
field, in our
Adaption
opinion,
dieser Technologien
in
voranzubringen.
order to
Ja, vielen
drive the
Dank
adaptation of
für eure
these
Aufmerksamkeit
technologies.
und
So I'd like
hoffe,
to thank you for your
dass
attention,
es nicht zu schnell und
and I hope
zu komplex war.
it wasn't too
Vielen Dank.
fast nor too complex. Thank you.
Wunderbar, danke Christian.
Thank you, Christian.
Sehr spannend, vor allem beeindruckende
That was very
Präzision.
exciting, particularly the reduction numbers in
sind
the
jetzt
number
ein
of
bisschen
animals
hinter der
killed.
Zeit, aber wir
We
haben
are a
eigentlich
little
generell
behind
genug Puffer,
schedule,
deswegen auf
but we
jeden Fall
still
noch
have
2
enough
Fragen
buffer,
an
so
dich.
we have time for two
Und zwar
questions.
einmal, gibt es schon Substanzen, die nach einer
Are there
Auswertung bei
substances
euch
already that after an
in
evaluation
Studien
at
gekommen sind
MPSlabs made
und
it to
haben
clinical
die ähnliche
studies? And did
Werte
they deliver
ergeben,
similar
wie
values
die von euch vorhergesagt
as
wurden?
those that you predicted?
Genau, ja. Also
Yes.
die kurze Antwort ist ja.
In a nutshell,
Wir haben
yes.
sozusagen mit Klienten schon zusammengearbeitet.
We already
Wir
worked
waren
with
eben auf der Folie zu
clients.
sehen.
You
Wir haben
saw them on the
tatsächlich
slide.
genau präklinische Studie gemacht. Wir haben
And they did
Daten von
preclinical
einer Handvoll
studies.
Medikamente aus deren internen
And we
Pipeline
used
bekommen, haben die
data
analysiert und konnten
of some drugs
sozusagen
from their internal
das
pipeline. We
hands-on
analyzed
vergleichen zu
them
klinischen Daten, die die haben. Das war
and we
sozusagen
could compare
eine
them to the
blinded
clinical data, but it
prediction.
was a blinded prediction
Und
actually.
genau, das
And
funktioniert
it works.
ziemlich gut.
It works quite well.
Okay.
Great.
Und dann ist noch eine
And then there's another
Frage, das ist ja in
question,
letzter Zeit sehr großes
which
Thema,
was
die
a very
Virtual
topical
oder
topic
Virtual
recently,
Control
the
Groups
virtual
für die
control groups
Pharmaindustrie.
for
Da heißt es ja auch,
pharma.
man kann im
And they
Prinzip ab
say
sofort bis zu 25%
that
der
up to
Tiere einsparen.
25% of the animals,
Wie ist das
the
mit
number of
diesen
animals killed would be reduced by
virtuellen Kontrollgruppen?
25%. What about the
Kannst
digital
du
control
da noch mal was
groups?
zu sagen? Wäre das auch etwas,
Can you say
was
something
wir
about
quasi anwenden
that? Is that something you would
Genau, es
also
sind sozusagen
apply?
zwei Sachen. Also aus Sicht
Two
des
things.
MPSlabs,
From the
wenn wir uns auf die
MPSlabs point of
Mikrosysteme
view,
konzentrieren, können wir
the
quasi
systems we focus on, We can simulate control groups. We can take away parts of the experiments. We are complementary to the biological experiments that they carry out. And we can say, for instance, you can carry out less bio-experiments because we can predict part of them, or you need to take fewer measurement points to record a curve, because we can tell you when you should measure so that we can extract the maximum of information, and this will lead to complementary information, which is at least as predictive. From the point of view of SQLabs, we did that already, clinical trial simulations with hundreds or thousands of virtual patients you used to simulate body weight, size, different parameters that you vary. And then you look at the distribution. What would it be at the end of the day? And we did that already for some customers. And they then used the information and went to the FDA or the EMA to file for approval.